All right, so we are live. Trina, you ready to go?
I am. Can you
All
hear me
right.
okay?
We can hear you perfectly. All right, so
Excellent.
I'd like to welcome everybody today to Micro Seminar for December 2016, the last one of the year. You might have been following on Twitter, but we have a whole schedule packed for next year, and we're pretty excited because there's some really awesome talks, and we actually have a 50-50 gender balance. So we were shooting for that, and we made it. So thank you, everybody that's volunteered. We're going to have some great talks coming up. So before next year, though, we're going to have Trina McMahon today. So Trina McMahon is she said she was traditionally an engineer. She's been engineering all her life and now is doing bacterial stuff. So Trina started as she's an engineer by training. She started with a bachelor's at the University of Illinois in Urbana-Champaign, working with Lute Raskin. So she did her undergrad there, did her master's there. This is actually the second micro seminar in a row with Raskin Link. So Amit was working with Lute as well. So we should probably have Lute on eventually. So after her master's, Trina went to UC Berkeley and got a PhD there in Jay Kiesing's lab, quote unquote, before it became huge. And in 2003, she started at UW-Madison, University of Wisconsin-Madison. UW to me, I'm an Oregon duck, so UW is always Washington, but but she started at University of Wisconsin-Madison in 2003 and has had a lab there ever since. Little story, just as how I came to know of this world. Like I said, my training is completely different than other people's that run micro seminar and kind of a lot of other people that have gone before. So I first learned of Trina's work because I think she had or Jesse might have stolen it or whatever. Jesse Shapiro used her data in a paper about 1.5 years ago. And it was an awesome time series of bacterial genomes and just following bacterial genomes and populations over time. And that's when I first kind of became aware of the McMahon lab world. And it was awesome. And I started looking into her papers, and it's just a great body of work. And so we got her here today to give a micro seminar. I think it's going to be great. And so without further ado, Trina McMahon.
Awesome. Thank you so much. It's very fun to be here. I think this is just an amazing thing to do for the community. So thanks to all of you guys who are doing all the hard work of organizing and making it happen. So I'm assuming that everything is good. You can come back on and tell me no if it's not. All right. So I'm Trina McMahon. Sorry. My startup disk is almost full. That's a bad sign. Oh, my computer survives this. All right, so I'm Trina McMahon. I'm at University of Wisconsin-Madison, and I'm going to tell you today about some work that I've been doing pretty much since I arrived. I'm really fortunate to have amazing collaborators here, and also through the Joint Genome Institute out in California. And so a lot of the work that I'm going to be talking about relates to work that we do with them. And I'm a huge proponent of JGI's community sequencing program, and really encourage everybody to take advantage of that and put in proposals for all of your awesome projects, because it's a really great way to get sequencing done for free. So I'm going to tell you today about work that we do in my lab. This is our lab logo, created quite a few years ago now, but I like it because it captures sort of the full range of the water cycle that we study. So I have a full, half of my lab working on wastewater treatment processes, and mostly we study things like phosphorus and nitrogen removal. And that, again, goes back to my engineering training. And I'm not going to talk about any of that today. I'm mostly talking about freshwater lakes and the bacteria that live in them. So we study how those bacterial communities change with time and through space. We're really interested in the processes that they carry out, so their functions, like their nitrogen cycling, their carbon cycling, their phosphorus cycling. So all of the same things that we care about in wastewater treatment, but in a much more wild kind of ecosystem where we have almost no control over them, but we have really great observational opportunities through our North Temperate Lakes long-term ecological research site. So mostly focusing on bacteria and lakes. And so these are some of the big questions that we think about in my lab lately. And I think, I don't think we're unique in choosing some of these questions. A lot of these are ones that many microbial ecologists are pondering right now. So we think a lot about whether we're looking at the right taxonomic unit, whether we're looking at one that's ecologically relevant to the question that we have. So we're going to be talking quite a bit about that today, actually. And that has got us thinking now about drivers and constraints on diversification of microbial lineages. And so that's also going to be a focus of what I talk about. I'm not going to talk too much today about predicting community dynamics, but I think many people in microbial ecology would agree that this is sort of our holy grail. I think not everybody has a clear understanding of why they might like to be able to predict community dynamics, but almost everybody wants to. I think we have the sense that if we're able to do that, then that means that we understand the system well enough that we can then proceed to ask even more sophisticated questions. So I'll talk a little bit about that, but not very much. We also are really interested in trait-based ecology. So how can we assign specific traits to coherent phylogenetic groups that we have in that we see in the data sets that we're generating from our time series of our lakes. And then ultimately, of course, we want to be able to look at ecosystem level processes from genomic data and sort of functional genomic data. So, and I'm not going to get into that at all here. That's something we're still really working hard on, but it's not really ready to talk much about. So I have two vignettes. The first one is going to be about this first question of what is the ecologically relevant unit to think about when you're studying microbial communities. And we're going to be using a time series of metagenomes from Lake Mendota, and I'll tell you more about Lake Mendota in a moment. And we have thought a lot about how we delineate populations and then track them through time. And that work isn't published yet. It's submitted to bioRxiv, but I've sort of been sitting on it and haven't gotten it submitted yet. So in the interest of not making people review articles over the holidays, we'll see if I get around to it before January 1st. The second one is published work and is the same data set that Jesse Shapiro did some work with as well. And that's looking at populations in a bog lake, and we are pretty sure that we observed a genome-wide selective sweep in those populations. And so we'll talk a little bit about how that relates to the ecotype hypothesis for diversification. So these are the two lakes that I'm going to be talking a lot about. So let me introduce you to them so you get a sense of place and what we care about as limnologists. So Lake Mendota is a eutrophic lake located in Madison. You can see the capital of Madison, or capital of Wisconsin here in the background, the Capitol Dome. So this is a photograph taken near the Center for Limnology. So it's not in the picture, but it's really just nearby, where we can actually go out on the lake in a boat straight out through a garage that's in the building. So we can actually get out on the lake really easily, and we take a lot of samples, sometimes more than once a week in the summertime, and study things like the cyanobacterial communities there, the heterotrophic bacterial communities, and we're really fortunate to have a lot of other limnologists studying the lake as well. Trout Bog Lake is pretty different. It's a dystrophic bog lake, so it's darkly stained like tea. It's smaller. It's in northern Wisconsin, and it has pretty different organisms living in it than live in Lake Mendota. I mean, there's still freshwater bacteria, but the bogs are slightly acidic, and so we get quite different populations, quite different lineages that assemble there. And I'm not going to talk much about the difference in that community composition, but just so you know, they're quite different ecosystems. Now, here's another picture of Lake Mendota. I like it because it shows the landscape around the lake. And so up here on the left is the state of Wisconsin, obviously not with snow, but if you look down here, you can see this chain of lakes. That's where Madison is. And then if you look from the north on Lake Mendota, you can see the city of Madison, the university, and then you can see that it's embedded in a landscape that is a mixture of urban and agricultural, and it's slowly becoming more developed. So it's a eutrophic lake, lots of cyanobacteria. We don't use it for drinking water, but it is used quite a bit for recreation, and there's a lot of concern about the cyanobacterial blooms and their effect on water quality, especially with toxin production. But not going to talk much about that today. But just to give you a context for the lake that has given us so much of this beautiful data. So I like to talk about this as a data smorgasbord. So I collaborate a lot with some Swedes, and I'll mention them at the end. we in that collaboration, along with work with Ramona Stepanauskas and Rex Malmstrom at the Joint Genome Institute, and also through the Earth Microbiome Project, we've done about 100 samples for 16S gene tag sequencing, but that's actually not very impressive anymore. When we first did that in about 2010, it was a big deal, but now it's just sort of your average data set size. What we're most excited about is this data set of time series shotgun metagenomes from 5 years. So it's about 92 samples. We have binned about 100 population genomes or metagenome assembled genomes from that data set. I'm not going to talk much about those today. Mostly I'm going to be talking about the single cell genomes that we have generated in collaboration with Ramonas and also JGI. So we have all this data, so we have all kinds of different ways that we can look at the community level dynamics, the genome level differences between the main lineages that live in the lakes, and sort of what that means for diversity as well as function. So let me tell you more about the bacteria that live in Lake Mendota. So this is from the ITAG, excuse me, ITAG time series, so about 100 samples. And this is work being done by Robin Rower. And the data were originally generated through the EMP. And so what I'm showing you is just a rank abundance plot of the percent of reads that are binned or assigned to specific clades of freshwater bacteria over that 10 years. So capturing what we think is at least the a full spectrum of the variation that we see seasonally in the lake, because it's important. If you just go out and collect a sample in July, that doesn't represent the sort of full complement of bacterial clades that you see across the whole year, because we also have ice on season, we have spring and fall being very different. So a lot of these names probably don't mean much to anybody else unless you study freshwater, but as I'll talk about more in a minute, you know, some of these top 10 clades are very cosmopolitan, so we see them in all lakes. In fact, these, 1, 2, 3, 4, 5, 6, 7, 8, the top 8 are detected in every single sample. So they're always there. Their abundances go up and down, but they are sort of what we call our persistent and cosmopolitan clades. And then you get, of course, the usual long tail, and I'm only showing part of it. But then there's other groups that are at different times of the year can be very abundant. What's exciting about this list, I mean, beyond just being an inventory of what's in the lake, we are able to relate, those to what we know about the ecology of many of these lineages. So quite a number of years ago now, Ryan Newton led a really ambitious project to create a field guide for freshwater lake bacteria. And so this is where we were able to collect up everything we knew in 2010, 2011 about all these different clades of freshwater bacteria. So that list that or that rank abundance plot that I showed you has, you know, a lot of those clades are represented in this field guide, which is about 30 pages long, 35 pages long. And so if you want to know anything about those clades, you can go and look them up and say, oh, here's what we know about what they eat or where they appear and when. And so that tells us a lot about sort of even just at a course level, what we might be looking at when we look at a particular community in the lake. So we are also very fortunate that either through single cell genomes or metagenome assembled genomes, we have representatives of all of those clades that are present in every sample. And then we have most of the other top 20, I think this is, or so. We're still missing a few. Although those, we may have gotten those since the last time I updated this slide. But basically it just says that we have reference genomes from these clades that we can use to think about you know, whatever we want to dig into in terms of the gene content or the heterogeneity within the population. So you might be wondering sort of what level of phylogenetic resolution this represents. The AC1A clade shares, so, you know, usually like 98% or greater 16S ribosomal RNA gene sequence identity across the full length gene. So these are very finely resolved groups. So just keep that in mind. So let me tell you a bit more about those top 2 clades. So the AC1A and the AC1B, and then there's also an AC1C down here and sort of more in the middle. So let me tell you more about these organisms. So these belong to a lineage within the phylum Actinobacteria, and Actinobacteria are very abundant in lakes. And it's largely because of these two major clades. So the A clade and the B clade. So all of these on this branch here are the A clade, and then these are the B clades. And so what I'm showing you here is the clade abundance, and that's abundance within that data set of all of the studies that had been conducted as of 2010, and all of the 16S sequences that were available at that time. So it was Sanger sequencing, of course, and so the numbers of sequences were a lot lower than what we have now, but in terms of the length and the quality of the sequence, we were able to definitively, bin those into different, what we call tribes. So there's the A1 tribe, the A2, A3, and so forth, and then the clade is A, and the clade is B. So you'll see that B1 is the most abundant in that data set of 16S genes that we looked at, and then the A1 and A6 are sort of the two runners up for most abundant. Now, prevalence refers to how many of the data sets were they observed in at all, and so again, you can see that B1, A6, and A1 are kind of the most prevalent tribes within that data set. So the bottom line is that these are really abundant, really prevalent. We call them cosmopolitan groups of bacteria that we see in all lakes. Up until a few years ago, we didn't know very much about what they ate or what they did. We know they're very, very tiny, so they actually will pass through a 0.2 micron filter. We now know that their genomes are very small as well, so they're on about the same size as Pelagibacter. So teeny tiny streamlined genomes, and everywhere, and abundant, sometimes up to 50% of the number of cells in a liter of lake water. So they're abundant, presumably they're important. So I'm going to get in now to some of the work that we're doing with Rex Malmström at the JGI, and this is also in collaboration with Sarah E. Garcia, who's now at Uppsala University, and my PhD student, Sarah Stevens. And addressing that first major question that we spent a lot of time thinking about, which is what is the appropriate coherent ecological unit to keep track of. So we are fortunate to have all these single cell genomes, and we have quite a few from that group of AC1. So this is showing you a pretty typical recruitment plot. So these first started appearing when I think it was around the time when the Global Ocean Survey was conducted by Craig Venter et al. And so it's this taking a reference genome and stretching it out. So the x-axis is the reference genome, and that's the coordinate on the reference genome. And then the y-axis is the percent identity of that metagenomic read as compared to the reference, and then each dot is just a metagenomic read. And so for those who care, we use some cutoffs to make sure that we were looking at reads that really should have aligned. And this is an AC1 single cell genome, happens to be from the B1 clade, where I've taken a Lake Mendota metagenome and compared it against that single cell genome. So the single cell came from Lake Mendota, and the metagenome also came from Lake Mendota. And they came from the lake at about the same time. It's not exactly the same date, so it's within a couple of weeks of each other. But so basically that single cell genome, the population that it represented should have been in the lake at that time. And what we see is that there are a lot of metagenomic reads that recruit near 100% identity, but then there's also sort of this smear down to about 95, and then there's sort of it seems like there's less points here, and then it picks up again down here around 85%. And then there's these other smeary places which represent regions of high conservation in the genome. So we can look at these plots. The problem is it's hard to look at these when you have 90 samples, and then maybe you have 10 or 15 different single cell genomes, SAGs that you want to compare them to, and so it quickly becomes not the right way to look at the data. So if you turn this on its side, and now we look at the percent identity on the x-axis in bin, so this is like a histogram. So we're looking at the frequency distribution of how often metagenomic reads are having that level of percent identity against the reference genome. And so there's your many reads that are matching at a close to 100%, and then there's that big cloud that's at more between 70 and 80 and to 90%. And again, this is the AAC1B1 genome that came from Lake Mendota, and the metagenome that we're comparing it to is collected at around the same time as the SAG. Now, what we see, first of all, that catches your eye is the fact that there's this dip here in the coverage or in the amount of reads that are recruiting to the reference genome or mapping to the reference genome at this level of sequence identity. And we call this a sequence discontinuity. And we certainly are not the first to observe this. Kostas Konstantinides has been thinking about this for ever since he's been in the TG lab and still today. And he talked quite a bit about this with pure cultures, where you can actually compare genomes from one strain to another and look at the same kind of discontinuity and sort of frequency, a large frequency of genes, or sorry, regions of the genome that match with high identity and those that are less, still probably homologous regions, but with less percent identity. So we decided that this was a nice way to think about the populations that we are catching with a SAG. So if we think about going fishing in the lake with a SAG genome as a reference and trying to find what populations we can pull out of the lake, bioinformatically in the metagenome, and then looking at those populations more closely. Now, the first thing we realized was that if we have a SAG from a different lake, it does a poor job of capturing the population that's present in the lake that we have the metagenome from. So here we have a SAG genome that's coming from Damariscotta, which is a lake in Maine. This was one that Ramonas Stepanauskas sampled. And the first thing you should note is that although we get a decent number of reads mapping, we actually don't get those very nearly identical reads up at 100%. And so that SAG does not represent the population that we see in Lake Mendota at that time point in June of 2009. If we take a gene, if we did this with a bunch of different SAGs, and we were surprised to find that in some cases we did see genomes from other lakes doing a good job of capturing reads from Lake Mendota. And this is one of the bigger surprises, which is a SAG from Lake Stechlin, which is in Germany. And it actually does recruit above 90% pretty well. But you'll also see that there's kind of this shoulder here, and it doesn't sort of go up like it did with the Lake Mendota sample. So here's your Lake Mendota sample that sort of has this peak density of reads at around 100%. There are a few reads that are at 100% that are being recruited by the SAG from Germany, and then none at all in that range from Maine. So it got us thinking quite a bit about how the reference genome is influencing our perception of the population in the lake that we're studying at that time, because you can only see what matches your reference. And so that's a good thing to keep in mind if other people are using this idea of mapping metagenomic reads onto a reference. If the reference isn't a good representative of what's in your ecosystem, then that mapping isn't going to work very well, or you're going to be missing a lot. And so we think a lot about that and trying to figure out what that means, both in terms of sort of the genome evolution and the history of the organisms, but then also how that affects our ability to actually pull out the populations that we're interested in. And so then it can get even more interesting, because here now we have I have 4 SAGs, one of which, the orange one, is from another lake in Wisconsin that happens to be oligotrophic, so it's not as green as Lake Mendota, but it's also in northern Wisconsin, so it's farther away, quite a bit farther away from Lake Mendota. not as far as Maine or Germany, but we can see that there's also lacking that shoulder, or that it drops off here as we get close to 100% identity. But then we also have 3 different SAGs from Lake Mendota that even have slightly different shapes. So this L06 and A23 lay almost on top of each other, but the J17 has more reads that are more different from the reference, or more divergent from the reference, which suggests that it's part of a different population. I'm not going to talk too much about the phylogeny of these organisms. If you're interested in that, you can look at the paper that we have on bioRxiv, because I wanted to make sure that I have time to talk about some of the other very cool stuff. So, so the next question we wanted to know was, how does this look over time? Because this was the metagenome, a metagenome that was collected around the same time as the SAG. So does it continue to look like this as we move forward in time? Or do we start to lose those reads that are recruiting at that very high percent identity? What does the discontinuity look like? But first, I want to take just those reads that are mapping or recruiting at above around 95%. So this green shaded part is, now I'm going to take that region and I'm going to look at those reads, and I'm going to look at them over time. And so this is kind of a busy plot, but just let me walk you through what you're looking at. So the dates are on the x-axis, and we have now pooled metagenomes by month. So we sometimes have 2 or 3 or 4 metagenomes for each month, but we pool them in order to get enough coverage for each one, and also just to make it easier to look at. We know that bacterial communities are variable through time on timescales of probably like weeks in lakes, especially in the summertime, but it just gets easier to look at by month for this purpose. So we have 5 years. And here we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 single cell genomes from the AC1 lineage, representing different clades and tribes. And the circumference, or sorry, the diameter of the spot is related to the abundance, or the relative abundance of those reads in the metagenome, but the absolute number doesn't mean a whole lot. It sort of is loosely related to coverage, but not perfectly. But certainly comparing within the circles, you should be able to say that big circles are more, means that that population was more abundant than small circles. The color is related to the average nucleotide identity. So it's taking this part of the frequency distribution plot, cutting it out, and calculating the average nucleotide identity of all those reads. And so first, what you should notice is that the SAGs that come from Lake Mendota all recruit reads that are at a higher percent identity than the SAGs that come from this other lake, there's Sparkling Lake, which is the one in northern Wisconsin, and then Lake Stecklin in Germany. So we get population, the SAGs from Lake Mendota are better representatives of the populations that are in Lake Mendota than SAGs from other lakes, which I kind of already made that point, but this just shows it through time as well. But you can also see that there are sort of blooms and busts of some of these populations. So the gray circles means that they drop below our detection limit. So this particular population that's caught by this SAG and from the A6 tribe, is really abundant in this case in April, but then it's gone in August and September. And so that sort of plays out each year that we have sampled. Then you'll also notice that even for the ones that are present all the time, you kind of see this pulsing dynamic. And so this, we can look at this a lot and scratch our heads about what could be causing some of these differences, and I'm going to go through a little bit of them. I mean, okay, so this is the first thing that I mentioned, which was that the not from Lake Mendota has lower percent identity. The other point I wanted to make is that these 2 SAGs represented by L06 and A23 themselves are nearly, they share very high average nucleotide identity with one another, so 99% or greater, and or greater than 99%. And we think are members of the same sequence discrete population. So we pulled two cells that belong to this sort of operational definition of a population when we sampled the lake for those single cell genomes. We don't have very many examples of that. Usually when we have two SAGs, they do not represent the same sequence discrete population. So it's actually quite rare to be able to get two of them. So for example, we're pretty sure that this J17 does not belong to the same sequence discrete population as L06 and A23. And you can also see that that's reflected in the fact that they have really similar dynamics and similar average nucleotide identity. One really interesting thing that we didn't notice until we really stared at this plot for a long time is that the differences, there's actually a pretty striking difference between the average nucleotide identity of the population from 2008 to 2011 in those two, when using those two reference genomes. It drops significantly in 2012, so that actually is a statistically significant difference. And another way to look at that is just to look at the, again, the average nucleotide identity, but sort of zooming in now between 99 and 94 or 95%. And in the years 2008 to 2011, the population that was present was sharing about 92 or 99.35 or 99.4% identity with that genome. This is the L06 genome. And so the blue to greenish colors are all from those years. And then the pinkish ones are from 2012. So you can see that it did drop and that this difference is significant. So we dug into this a little bit more, trying to figure out whether or not it was a new strain that had, a new subpopulation that had arisen in the lake, or if the population itself had just become more heterogeneous, so more variation within the population. So we looked at the number of variant sites when we mapped the reads, and across both those blocks of years, we had the same number of variant sites. So it's about 6.3% of the sites are have SNPs in them. So single nucleotide polymorphism, so differences between the reference genome and the reads that we're mapping to it. But in 2012, more of those variant sites have mismatches to the reference genome, right? And what it looks like is that this population hasn't become more diverse within itself, but that it's just a new strain that has different bases, different alleles at those variant sites. So that's interesting to think about why that would be. Does it matter? Is it going to continue forward if we continue sequencing, to current day, and wondering what might have caused that shift, because before 2012 it was actually pretty consistent, right? So the cells that we sampled in 2009, all of these single cell genomes were collected in 2009, did a good job of representing the population in 2008 and going forward until 2012. And so, now we're very interested why that would be, but we actually don't have an answer at this point. So we're still digging into that. Now, the other thing we wondered was whether or not this behavior or these patterns that we were seeing were something that was specific to the AC1 Actinobacteria, or if it's a generic or general property of freshwater bacteria or any bacteria, or maybe just aquatic bacteria, we don't know. And so we took other single cell genomes that we've recovered from Lake Mendota and did the same kind of analysis, and most of them did look similar to the AC1, but one of the big outliers is this group called LD12, which I believe is now in culture from Cameron's lab, which is pretty awesome. But it's enigmatic in the same way that AC1 is, and that it's very tiny, very streamlined genomes present in many lakes in high abundance, and we don't really know too much about what they do or how they live. And they seem to be quite the exception to that observation of the sequence discontinuity. So what we have here are 5 different SAGs, all from Lake Mendota. You can see that some of them have that peak near 100% sequence identity, but then the drop off or that sequence discontinuity is much less pronounced, and it's almost and then there's no big hump at around 80%. It's almost like that 80% hump that we saw for most of the other SAGs is sort of smooshed up against the 100% hump, which makes us think that the populations of LD12 haven't had as much chance to diverge from within each other as compared to things like AC1 and some of the other major lineages and lakes. You know, other indications are that LD12 is pretty special, and there's some work that our collaborators have been doing in Sweden, Siv Anderson and her group, looking at genome evolution of LD12 and in the larger SAR11 clade, because LD12 is part of SAR11. It's closely related to Pelagibacter, but it's quite distinct from Pelagibacter as well. And so it's really interesting to think about why LD12 is different and what that means for its ecology, and also its both past and future evolution. So we also wanted to think about how that relates to the abundance, the changes in abundance through time of these different populations. So in the top panel, you see the AC1. So those are the different SAGs. Remember that A23 and LO6 are part of the same sequence discrete population. You can see they track each other pretty well. Now this is just relative abundance as inferred by mapped metagenomic reads. So it's a pretty, that's a pretty, I can't say that it completely reflects the actual abundance of those populations, but it's another proxy that's, I don't know if it's better or worse than 16S tag sequencing, but it's certainly more finely resolved at a phylogenetic scale than 16S sequencing. And the red one is J17. It was that population that had the larger circles on the bubble plot that I showed you. But you can see that they have quite different, some of their dynamics look similar to the other populations, but others are quite distinct. Then you have this I14, for example, which sort of has a few bursts of dominance, but are not nearly as dominant most of the time. So interesting different dynamics. When you look at the LD12 SAGs and how the reads that they recruit change through time, you can see much more synchronous dynamics. So when they're high, they all go up. When they go up, they all go up together. When they all come down, they come down together. And that suggests that there's more, there's less ecological difference among those populations, because if they're all responding to the same environmental forcing or interactions, then that could explain some of those synchronous dynamics, whereas the AC1 SAGs are behaving more differently. So that means that could be something very interesting about LD12 versus AC1 as well. So just a few lingering questions that we're still scratching our heads about, and trying to figure out sort of how common the sequence discrete population phenomenon is. So how often do you see that discontinuity in the frequency distribution plot? And why do they not exist sometimes? How is that influenced by the reference genome that you're using as well, is an interesting question. I would love to think more about the lifestyle of the organisms and how that relates to the population level heterogeneity. So we know the LD12 and AC1 actually have very similar lifestyles. They're small, they tend to be grazing resistant, they have streamlined genomes. They seem to eat pretty different stuff just from looking at their genomes, and also some work done with fluorescent in situ hybridization by other groups. But trying to think about how those differences would play out in how the organisms actually make a living. And then wanting to figure out why certain populations might shift in terms of their percent within population percent identity, and why certain strains would arise at particular times and not others. So that's the end of my first vignette. Don't worry, my second one isn't as long. But so hopefully I convinced you that thinking about this ecologically relevant unit is important, and that we think that we've come up with a good way to delineate sort of this, delineate populations within this this continuous smear of metagenomic reads that come out of a recruitment effort like this, where if you cut it off at 95% and look at the reads that are above that, that's a population, and that anything below it is a member of a different population. And so excited about being able to use of what you recruit with specific reference genomes as a way of tracking those populations through time. Of course, that also tells you that you need to work pretty hard to get as many reference genomes as you can from the environment that you're studying, sort of from the ecosystem that you're interested in pretty specifically. And I don't think we're anywhere near that with even Lake Mendota with the hundreds of genomes that we have. So we're still trying to work through that. So the second story I want to talk about is work that we've also done with Rex Malmstrom. And it sort of addresses this question of how do lineages come about, or distinct lineages come about in these ecosystems. And this was with Matt Bendall, who at the time was working at the JGI, but now is a graduate student, and also with Sarah. So maybe you're familiar with these two sort of competing, contrasting speciation models for microbes. Some of this was originally, a lot of this was originally developed by Fred Cohen, but it's also been put forth by Jesse and his mentor, Martin Poltz. And so to contrast them, it's just that in one model, there's more recombination occurring within a population than selection. And so that would lead to a particular SNP being selected for and becoming fixed in a population where there's diversity within the rest of the genome, but that's actually the gene or the SNP or the that specific locus that's being swept in or selected for in an event. Now, in an ecotype model, you have less recombination than you have selection, and that you can actually see a full clonal sweep of a genome where the whole, some part of the genome is under selection, but then that entire genome sweeps the community and expands to fill, you know, a niche or a new niche. And so most people think of these as being in competition with one another, and as a model for evolution, I think everybody is, when you get down to it, willing to admit that it's probably some combination of those two, and it may be that certain lineages follow one model or the other, or that under certain situations you would see one model playing out versus another. But everybody's very interested in finding evidence for one or the other in actual real wild ecosystems. And so this is just another way to emphasize that under the recombination model that you have diversity within the genome, whereas in the ecotype model you see homogeneity in the genome after a sweep. So it's a genome-wide sweep as opposed to a gene-specific sweep. So thanks, Jesse, for making that so clear in your paper. Even I can understand it. I'm not trained as an evolutionary biologist. Let me just disclose that right now. Oh, that's kind of a problem. Let me pull that back up. Pardon our technical difficulties. Hopefully this works. Or else you can't really do a chalk talk in Google Hangouts, can you? All right, so now I'm going back to Trout Bog Lake. Remember, I mean, for what it's worth, I'm not sure how much the differences between the two lakes actually influence the different data sets that I'm showing you, but I was talking about Lake Mendota exclusively before, and now we're going to be talking about Trout Bog Lake, which is in northern Wisconsin and is quite different. So we actually noticed this sequence discontinuity that I mentioned in my first little story when we were looking at metagenome assembled genomes from Trout Bog Lake, and we saw that when we took all those mags that we assembled and mapped the reads from Trout Bog back onto it, we saw this extreme sequence discontinuity. But again, we're looking only now at the reads that map near 100%. And we looked at, you know, more than 30 genomes, but 30 genomes is what ended up in the paper that we published. Was that last year or this year? I can't remember when it actually came out. Oh, it says it's in review. It's actually published in ISME Journal now. Sorry, I didn't update that. So we looked really closely at those 30, and then we looked even more closely at a handful that were very interesting. But before I show you those data, let me walk you through how we represent the data. So what I'm showing here, and this is more of a cartoon, this is not actual real data. So this is looking at individual single nucleotide polymorphism loci. So, and at different time points. So let's look at time point number 1. So at time point number 1, we look at all the metagenomic reads that map onto a genome, a reference genome, and we look at particular positions in the genome, so a location in the genome. So at a particular base, at time point 1, 25% of the reads have an A in that location. 75% of the reads have a T at that location. As you go through time, that can change because more reads will have an A. Here more and more having an A. Now it's almost 50-50. Then it went back down again. So you can have sort of varying amount of heterogeneity at that variant site through time. Now here's another variant site where over time it actually becomes more and more and more G and more homogeneous to the point where all of the reads in the metagenome that we mapped to our reference genome are 100% identical to the reference genome. Here you have 0, so in this case all of them have a T at that location, and so on and so forth. So the color represents the extent to which it's homogeneous between 0 and 100% different compared to the reference, okay? So this is all in relative to the reference genome, so to a particular mag. And so most of the genomes that we looked at look like this. So here we have 6 years of data from Trout Bog. And so the heat map that I'm showing you, these 2 heat maps are for 2 different reference genomes. And if you care about the names of the organisms, these are the groups that they're from. And the SNPs are just ordered here by their homogeneity at the first time point. So here we've pooled reads by year just to make it easier to look at and also to get more coverage. So about 8,500 SNPs in that genome, a lot. And so we can also look in a more summary way at the fraction of those SNPs that are dominated by a single polymorphism or a single allele, and see that that doesn't really change appreciably through time. And when you look at it, you don't see any pattern. It just looks scrambled. You're not supposed to see any pattern here, right? So this is what most of them looked like. But then we stumbled on one that looked quite different, and there's actually more than one. There's another one that there's another very interesting one that happens to be from TM7, but I'm not going to talk much about that. So this is a chlorobium genome, about 3,000 SNPs. And what fascinated us was that at the beginning, sort of in 2005, there were, sort of this many, that matched at about 100, that matched the reference genome not at all. And then there's some that match the genome perfectly, but then there's this stuff in between. And over time, we found that 100% of the reads eventually mapped to the genome. So what that tells us, the reads that mapped to the genome were 100% identical to the reference genome. So what that told us was that, initially there was heterogeneity in the population as compared to the reference, and then that heterogeneity was purged. And so you can see that in another way where you can look at the fraction of SNPs that were dominated by a single allele and how that went dramatically up in the latter half of the data set. And so what we think is that we observed a genome-wide sweep, a purging of diversity, where that diversity that was in the population here is gone by 2013. I should point out that when we first did this, we only sequenced up to 2009, and nobody believed us that this was actually something that would persist. And so then we went and we sequenced 2012 and 2013. And if you look closely, you can see there is some heterogeneity in 2012, but then more of it goes away in 2013. So we actually had a, for at least as long as we have now sequenced, that strain that swept the population is persisting. So it would be super nice to go back and do 2014, 2015, 2016. We can get some money. And we looked a little bit at more closely at the genomes that were gained, and the genes were gained and lost during that genome-wide sweep. And we did see that there were some genes that disappeared from the population and some that were gained. And so there was a genome content difference, not just a diversity or gene content difference rather than just diversity at the SNP level. And of course, the first thing we did was want to go in and look and see, you know, what are those genes? What made it sweep? Because this implies that these genes that were gained gave this strain an advantage, and so it could sweep the population, purging all the other diversity. Or maybe there's something about the loss of these genes that somehow gave this strain a fitness advantage. So you look at what those genes are annotated as, and unfortunately you don't get a lot of insight. You know, it's like many things, you know, you think once you get the annotation, everything will be revealed. Well, you know, there's a few interesting leads here, but nothing that we could pin to a particular explanation for why that genome swept. So if anybody has ideas, if you recognize, you know, any of these annotations as being important for something that you can think of that would give it an advantage, let me know. We haven't really been able to come up with anything. So we kind of had to leave that story as a, wow, here's a really interesting phenomenon that we observed, but we don't have a really solid explanation for why it happened. And so the bottom line is that we think we observed a genome-wide selective sweep, which, you know, not very many people, if anyone, had documented that in wild populations. So in a community that was, you know, out in is self-assembled in an ecosystem where you can actually observe the sweep happen in real time, right? This wasn't going out and collecting isolates and then going back and trying to infer what happened. It was actually watching it happen through time. And so we think that this provides evidence for the ecotype hypothesis, where we see clonal sweep at the genome level. I should point out or remind you that that was one genome out of 30 that we looked closely at, and actually out of hundreds that we had data for. So I'm not saying that this is a very common occurrence or one that's the rule. It may well be the exception, but it certainly is evidence that it does happen. And it's interesting to think about the lifestyle of chlorobium and what that might mean for the fact that we've observed it in a chlorobium and not any of these other lineages. So that's more food for thought. So that brings me to my acknowledgements. And I want to make sure that I say thank you, Rex, because he's been an awesome collaborator at JGI. And of course, we also work with Tanya Woike and Susanna Tringe as part of that, and now also Adam Rivers. Ramonas has been incredibly generous in terms of helping us with the SAG sequencing. The Earth microbiome folks had faith in us very early on in terms of doing a lot of our time series 16S sequencing. And then Stefan Bertelsen is a long-term collaborator in Uppsala, and Sara E. Garcia as well. And then there's some other McMahon lab members here. I didn't list them all, but these are the ones that work mostly on our lakes. And these are our funding sources and other partners. I'll also give another shout out for the Center for Limnology and the LTER here at Madison, who lots of amazing people working there, even if we often have to jump up and down and try to convince them that microbes are worth studying, because mostly they look at fish and stuff like that, but we'll forgive them for that. So yeah, with that, I'll take questions, and thanks again for giving me the opportunity to do some blah, blah, blah about stuff that I think is really exciting. Thanks.
Awesome. I'm going to monopolize question time while I search through.
So wait, do I undo my screen sharing now? Does that matter?
Yeah. Let's see here. Let us show the world.
Let me figure out if I can do that.
Hey, Dave, can I ask you a question? Because I got to go in 5 minutes.
Go for it, man.
And I think it's probably a quick one.
Oh, there we are. Hey.
Dave, you good with that?
Shoot.
Yeah, go.
Okay. So Trina, this is awesome. I love seeing the whole progression of the data over those wonderful time series. I'm curious about this dip in the recruitment plots in between the big hump and then the little hump, because I've seen that before too in marine SAR11 recruitment plots that we've worked on. And I was just wondering, you know, someone asked me this question when we first had that data, and I still don't have a great answer for it. But why do you think there's that dip? Why isn't it just a gradual, like why would there be any preferential pattern there at all that's so reproducible? And why isn't it, say, like a steady drop off or a steady drop off this way or something along those lines? What do you think that's all about?
Yeah, and Rex thinks about this a lot. I wish he was online. My student Sarah, who I'm not sure if she's watching or not, but so the idea is, I think that there's something holding the population together, and there's that no man's land of sequence identity where it is almost like a biological species concept, right? Where you can't have gene flow or recombination, although then that implies that the recombination model is the important one, that would allow there to be a strain that exists in that no man's land. It's like it's like you either are able to recombine and interact with in the sense of gene flow with the things that are really highly related to you, but not to everything else, and that there's forces that are pushing those apart so that you're not having gene flow between them, so then their homologous regions aren't as similar anymore, right?
Yeah.
And so that's where we have this idea that, and it's largely Sarah who's come up with this, that the LD12 are just not as far along on that diverging path as the rest of the other ones. And why that would be, we don't know.
It still has that dip, though. So what I mean,
it has a little bit of a dip, yeah.
Even though the ones that have gotten out of there still, I mean, there's definitely, yeah, I totally agree with you that it looks like the populations in general aren't as different. But that initial little jump that you have to make to get out of, I guess that's a cool idea to think of, maybe using that to define what a true species is.
Yeah, or where if there's gene flow among them at least, or like the thing that bothers me sometimes with that way of looking at it is that it's taking a lot of important information about where the heterogeneity is, or where the variation is in the genome, and smashing it into one number, right? It's like the average nucleotide identity. It's just like, so, how much of the genome is shared? Where are the SNPs in the genome? So that's all getting lost. And so sometimes I struggle with whether, how much are you missing when you look at it that way? But it's certainly an interesting way to look at it.
Yeah, maybe a third dimension's needed that shows sort of distance along the genome or something like that.
Yeah, somehow. Or like, yeah.
Well, thank you very much. That was wonderful. I'm going to have to head out, but I really enjoyed it.
Thank you.
Cheers.
Well, all right. So I haven't gotten any questions from Twitter yet. Anybody else on the Hangouts want to chime in, or?
Yeah, I'll wait. I'll chime in. Do you hear me? Oh, wait, I'll turn my video on too. There we go. Hey, Trina, it's Jesse.
Hi. Yeah.
Nice to virtually meet you.
I know, finally.
Yeah, very cool. I'm at home on childcare right now, so I was like slightly distracted.
I know the feeling.
I enjoyed it very much, and definitely very, very cool stuff. Yeah, I wonder, I was also trying to wrap my head around the first part of the plot, where really within that, you're looking in that 95%, and there's some variation in about, within the 95%, how similar are you? So I think, yeah, I mean, that's, it's always been surprising to me as well, how, in different systems, different metagenomes, there's always that big gap between the clear discontinuity. But then within the 95%, what's going on there, right? So are those species on their way to drifting apart? Or, I mean, can you consider that like drift within a species where there's some genetic diversity there, but ultimately it ebbs and flows, and then always ends up sitting in that bin, even if it kind of diverges a bit, but then it kind of gets pulled back by. So, I mean, is that
that's how I originally was thinking about it. So the reason we made those bubble plots was because I had this idea that it was kind of going like this, where it would get diverse and then it would come back again. It would get diverse and then it would come back again. And I wanted to see if that was happening. I think once we worked out all the glitches of like, how do you have enough coverage for each time point and the reference genome, and are you filtering it correctly in terms of how you do the mapping or the blast or whatever? And I think a lot of that pulsing that I originally saw in the first machinations of the data went away. So like, if you look across time at those bubble plots, it's pretty consistent. I mean, the abundance is changing, but the color is not. And and when I was looking at that really deeply, I was pretty excited about that big difference between 2008 to 2011 versus 2012, because I finally saw like there's a difference there, but it's not like this sort of which is what I thought it would be, or was hoping it would be, because that sounds really cool. So I'm not, but then to think about whether or not you are getting sort of a diversity accumulating and then maybe going away, or something directional, I think you have to start looking at the SNPs and where they're located, and more than just looking at ANI, right? I think ANI is masking all this other interesting information about how that process is going on. And so right before literally an hour ago, 2 hours ago, Sarah and I were like trying to dig into some of the data so that I would have something new and exciting to say at the seminar at this. And we got a little bit into it, but didn't quite make it enough to talk about it. So what we need is awesome visualization tools like Envio. We haven't figured out how to get Envio running our computers yet. Sorry, Baron, if you're out there.
I'll ask another question if there aren't others, but I don't want to monopolize. Are there other people in the queue?
I think we're okay. Go for it.
Yeah. Okay. So I guess my next question is about the second part. And I think you were pretty in your talk. You were pretty careful about not actually using the term a selective sweep to describe what was happening, which is is in the title of the paper, I think. And it's a plausible explanation, but I guess I wonder, do you think it could also be drift, or could it be transient? Could it come back? Yeah, I mean, I'm self-admittedly I have a strong selectionist bent, but to play devil's advocate, do you think that it could just be drift just due to bottlenecks and population size changes and stuff like that?
I mean, I suppose we can't rule that out, but and when we present that, the first question, especially when we present to limnologists, the first question is, why did the new strain come up? Like, what was the selective pressure? Like, everybody wants to know what that is. And we can't point to anything. And so if we had been able to point to something, then the story would have been a lot tighter, and maybe that title would have been more reflective of what we saw. But yeah, I guess we can't really say for sure. The other thing that hurts my brain to think about is those mags were generated by doing a gigantic combined assembly of all the time points, right? So the mag assembled because it was clonal or nearly clonal at those later time points. Or at least that's probably a large reason why it was so easy to assemble. And so when you're using mags as your reference, like, how does that skew how often you see that kind of pattern? I mean, obviously we don't see it very often, but like the issue of the reference genome and how that influences what you see when you do the recruitment is one that I still can't get my head around yet. And I mean, if we saw it with a SAG, then I might be more convinced that it's happening more. I don't know.
Yeah,
I don't think it really addresses the selection thing. It's more about like, what are we seeing and how common is it, and is it more a function of the kind of reference that you have?
Yeah,
I don't know.
Yeah. No, I was talking to Murin and Chris Quince, who are also there's a lot of methods out there now that basically have the same idea, which is you assemble your metagenomes and then you map your reads, right? And your sort of operational definition of a species is the thing that you assemble, and then your operational definition of strain variation within that are SNPs based on what you map. And that's great. It's very convenient. It's pretty much as good as we have. But I was asking these guys, I was like, well, it's totally dependent on your assembly, right? Like what you end up defining. So I think this is sort of just it's as good as we've got for the moment, but it's not really clear what that really means. And it's very dependent on the assembly. So it's kind of like the assembler is telling us what relevant units we should be.
Right. I mean, and it's interesting in the I told my stories in the reverse of the order in which we did them. So the trout bog stuff with the genome-wide sweep was all done with MAGs, and we said, oh man, like, is this just totally an artifact of MAGs, at least the sequence discontinuity part? And then we started looking at the SAGs, and we saw that you see it with SAGs as well, and we're like, well, you know, if we see it so often with SAG populations, then it's probably real.
Yeah.
Or it's not an artifact of the assembly. But there are certain groups of freshwater bacteria that we know are there by the recruitments to the SAGs that we never ever get in as MAGs. So we don't get LD12. LD12 doesn't assemble. And partly why it doesn't assemble is probably because it doesn't have that sequence discontinuity. There's other lineages that we don't have SAGs for, and we also don't have MAGs for, that we know should be there based on the 16S, so we don't get anything that looks like it's related to it. So, you know, I think people who only work with MAGs are always going to be missing the members of the community that have some amount of heterogeneity within them that makes it impossible to assemble them.
Mm-hmm. Yeah.
Which everybody probably says, yeah, I know that, but like, how much do you miss, right? So we've been trying to figure out, like, how good is our reference genome collection at sort of explaining all the populations that we have, because now we have MAGs and SAGs, and haven't really gotten a good handle on that yet either, like whether we have enough.
Still no Twitter questions. So I actually have, I'm curious, I'm always curious when I see other people's stuff and they go through JGI. So apologies if this is kind of an ignorant question from my part. So what motivates JGI to be interested in this stuff? Like, is it just like, how do you get them interested to look at populations of bacteria?
Well, this was a CSP that we wrote in 2010, and it was, at the time, it was one of the biggest projects that they undertook in terms of amount of sequencing. And I think we convinced them to do it because, you know, it was linked to an LTER, and it was about carbon cycling. And I wrote it with Rex. So it was so Rex approached me and said, I heard you have this great time series of samples, and like, let's do a bunch of sequencing and just see what we find. And so all that proposal was about carbon cycling and hypotheses about how, you know, the gene content of the community changes over the seasons because you have different stuff going on in the lake. And then Rex had a postdoc who was looking at the data, the first round of data, and he saw this chlorobium genome that seemed like it, you know, had the genome-wide sweep. And suddenly Rex got interested in genome-wide sweeps and the ecotype model. And at first I was like, Rex, this is an artifact. This is too bizarre. It's not real. And everybody we showed it to said it didn't believe it, didn't think it was real. And so then Rex really got into it, and he wanted to prove that it was real. And so he sort of went down this path with it. And so we didn't plan on it. And it was very much him driving that second story. The first story is more something that I've been driving with Sarah and Sarah E. But it also was not part of the original proposal, right? So it's an example of, you know, finding something cool in the data and chasing it. You know, we still haven't done much with the carbon cycling question. So it was very serendipitous. And the other thing is that it's a bit unfair because I've been doing a lot of stuff with JGI since like 2004. So I don't think that really influences the review process of the CSPs per se, but, you know, I have that track record of working with them, so maybe that helps get the projects through the door. But it's true that the projects are much more successful if you have a champion within JGI to help you do it. Absolutely.
Cool. I'm trying to figure out, Mike Henson says there's a question on YouTube that I'm trying to find out where.
Yeah, I don't have the YouTube open. I'm not sure.
All right. So your second SAG from the bottom sort of followed the pattern. Not only pulsing by abundance, but also by phylogenetic relatedness.
Second SAG from the bottom. Let me see if I can do my screen share again and see which one he means. I mean, probably in the bubble plot. It's funny, we have these affectionate terms for the figures. The bubble plot. Second, oh yeah, it does kind of Yeah, you're right. Okay. Let me zoom in so you can see it better. So this A6I14, so not only is it changing in abundance, but it's also changing in ANI. That I'm a little bit worried is a coverage issue, right? So when it's abundant, like when the circle's big, it's dark. So that means it's very similar to the reference. When it's not as abundant and it's light, it's tempting to say that that's a different population. And so it's, less matching to the reference. And so that would be that kind of like pulsing in and out, but I'd want to look at the coverage of those points and whether or not they're approaching that limit of, because again, this color reflects the average of all the reads mapping across the whole genome. And we all know that different regions of the genome have more or less divergence. And although you'd think that would be the opposite then, right? You would think that when it's in low abundance, only the very conserved regions of the genome would be recruiting reads. So you would think that it would be a darker color. Ooh, I don't know. That'd be worth looking into more. Yeah, there's like a million things like that when you're staring at the data that you're like, oh, maybe I should think about this. Cool question, yeah.
That's it. No more Twitter. All right. And if anybody else has any questions, speak now or forever hold your peace. Except you can contact Trina by email or Twitter or any other passenger pigeon, maybe, whatever means you may be. Although it's getting cold in Wisconsin, so passenger pigeon's probably not the best idea. So, all right, with that, thank you very much again, Trina, for a great seminar today, and we'll see everybody again in a month with 2017's micro seminar. So, thanks again. It was great.
Thank you, and thanks again for hosting. It's awesome.